Okay, this is the third and final slide deck for this module 3 this week. I went for the perfect threes, if you will. What we're going to be talking about today is sensory information, reflexes, how do we actually bring input from the environment and into our brain, and then how do we flip that around and send it right back out to our muscles. One of the first types of sensory organs, if you will, that we're going to be talking about is joint proprioceptors. So what joint proprioceptors do is they give consistent and constant feedback to our brain and our spinal cord about what position our joints are in, right? So they're going to be serving a protective mechanism to let our brain know where we are in space, how our body is moving around and interacting with the environment based upon joint angles and joint pressures and joint forces. Does that make sense? Like, we need to be able to get a input to our brain about the position of our joints as well as the amount of force that our joints are trying to apply. If we didn't have that, then we wouldn't have a governor, right? We wouldn't have this ability to essentially shut down muscle action, shut down joint action to protect ourselves. That would be very scary if you're trying to get away from a lion or a tiger or a bear, or evolutionarily, if we have no way to protect ourselves long term from compromising positions, right? So there's three primary types. I don't know if you can see my fingers or not, I'm sorry. There's three primary types of joint proprioceptors. It's going to be free nerve endings. There's going to be Golgi type receptors. Do not confuse these with Golgi tendon organs, which we're going to talk about. And then there's Pacinian corpuscles. So free nerve endings are exactly what they sound like. They're going to be the ends of neurons that are going to be located near the joints. They're going to be the most abundant, the most frequently occurring, and they're really going to be used at movement onset to make sure that the correct muscles are firing, to make sure that the joint is actually initiating movement. The next type of joint receptor, proprioceptor, is going to be a Golgi type receptor. Again, this is a Golgi type receptor, not a Golgi tendon organ. I'll generally refer to them differently. So these are going to be found in ligaments and in ligamentous structures around a joint. Same exact concept as free nerve endings, just a little bit different, right? Then you're going to have Pacinian corpuscles. Those are going to be in the tissues surrounding, so kind of the peri-joint area. And what these are going to be used for is helping to determine the amount of joint rotation that is occurring. So approximately how much flexion or extension, how much rotation of the femur is occurring in this specific position, or around the hinge joint of the knee, how much rotation of that joint has occurred, to make sure that our body and our mind know what it's doing. Now we're going to get into muscle proprioceptors. As you can tell here on the left, this guy is completely natural. He's never touched a weight in his life, never touched a drug in his life, and never used Photoshop either, right? Skeletal muscle is really cool functional unit for the body. It does a lot for us, and one thing that it really does cool is it sends signal from itself to the brain, and then the brain sends signal back to it. Most of these sensory structures are going to be called mechanoreceptors. And what mechanoreceptors do is they help kind of facilitate the mechanical action of the muscle. It makes sense, right? Like, the term follows the function. Two of the primary muscle proprioceptors for our concern are going to be muscle spindles and Golgi tendon organs. I've got to say it because I said it like 18 times on the previous slide. Golgi tendon organs are not Golgi type receptors. Got it out of the way. Cool. Muscle spindles. These are going to largely function as length determinants. They're going to let the brain know how stretched or not stretched, how long, elongated, or short a muscle is at any specific time. They're going to run parallel with the muscle fiber. In order to determine length, it makes sense that it will run along the length of the muscle, right? If it didn't run along the length of the muscle, it wouldn't be able to determine accurately how long that muscle was. I'm trying to say stupid things to get you to remember. I said so many L's, I can't even talk now. muscle spindles and Golgi tendon organs, or GTOs. So muscle spindles are going to provide the brain with information about length. Oh my God, how many times can you say length, Parker? They're really important. They're going to be innervated by gamma motor neurons. And why that's important is that as soon as the length changes to a point where the body's not going to be happy with it, the brain's going to instantly send a signal and say, you need to contract, you need to shorten, you need to change length now. If we didn't do this, then slack would become present in the system and they wouldn't be super accurate. Plus, it might put our body in a compromising position. Muscle spindles are what is responsible for the stretch reflex, also known as the myotatic reflex. This is the knee jerk test, right? You're going to essentially hang your leg off in a short sit position for my 4460 folks. You're going to tap the patellar tendon. When you tap the patellar tendon, it's going to have an immediate lengthening action on the quadriceps. And when that occurs, you're going to have a muscle, a sensory system put in place that is essentially going to cycle around, cause the quads to contract to change the muscle length because it just heard that it was too long, and it'll shorten up. And that's why you actually get that knee jerk, right? That's why you get the lower leg extending out. What about Golgi tendon organs? I made sure to highlight that these are different than Golgi type receptors. Well, these GTOs are largely responsible for monitoring the tension in, right? So if we think muscle spindle, L's are all over the place, length is all over the place, Golgi tendon organs has a T in there, so it's going to be tendon and tension are similar. It's going to be telling us how much force a muscle is contributing, how much tension is being built up into the muscle or into the tendinous unit. And they're located within the tendon. They're located in series with extrafusal fibers. So that means they run along the length of it in specific positions. And largely, these are going to serve as safety nets for us. They're going to prevent our muscles and prevent our joints through going through such a forceful motion that it might actually injure us. Something that's really cool is that this is a phenomenon that we can somewhat manipulate with specific amounts of training. So it's actually one of the primary reasons why you can get a complete shutdown of the system when you miss something like a really heavy back squat or a really heavy front squat. And by actually taking and training at higher loads, by training at high intensity levels, or even supra-maximally, you can almost inhibit this system some so that you can exert more force, so you can exert more muscular tension and be able to lift stronger or heavier amounts of weight and be a stronger individual. So in the last lecture, in lecture number 2 for this week, we talked about the divisions of the system, divisions of the neuronal system. And we talked about the somatic and the autonomic nervous system. Well, the somatic is going to be largely tied to motor neurons. And what that means is that they're going to control the muscular contraction of things. They're going to actually control muscles and in motor-based activities. So shrinking or contracting, better word. They're going to control contracting and lengthening of muscles. There is an alpha. If you go back a lecture, we talked about how there's somatic and autonomic nervous systems and about how the somatic nervous system really controlled the motor division of the body, whereas the autonomic nervous system really controlled the subconscious level things, the things that we need to make sure that are occurring in the background. Otherwise, our mind would be exclusively consisting of breathe, heart rate, breathe, heart rate, breathe, right? Those things have to happen on a subconscious level, whereas contractions of skeletal muscle can actually occur on a conscious level or also in a reflex arc. So the alpha motor neuron, we're probably just going to refer to them as motor neurons because it's a more common way of referring to them anyways, I think. is really going to be the primary system here. It's really going to be the primary neuron. And what that means is that the cell body of these motor neurons is actually within the spinal cord. So the axon runs out of the spinal column and runs all the way to the muscle that it's innervating. It's kind of cool to think about. And that means that these axons can be meters in length, right? Like they can be a really long length, because think about how far the bottom of your spinal column is all the way down to your calf. It's a pretty good distance. And that's what these somatic motor neurons are going to be doing. When that motor neuron actually gets to the muscle fibers that it's innervating, you will see that that axon actually takes and splits into several, well, it could be hundreds or thousands of terminal branches. And these terminal branches actually kind of spread across different muscles, and they're what's used to make sure a consistent contraction occurs. What this motor neuron in everything that it innervates is called is a motor unit. And that's a very important definition to learn. And that is, a motor unit is a motor neuron in every single muscle fiber that it innervates. If we remember the all or none principle, if we remember the trigger finger, right? Every single time that motor neuron fires, every single one of those muscle fibers is going to fire. Every single one of them is going to contract. So if we're talking motor units, how do we know the different types of motor unit? Luckily, to make it easy, someone decided to group them by size. So we're going to have smaller motor units, medium motor units, and large motor units. They can be as small as 20 to 1, even smaller, and they can be as large as 2,000 to 1. So that means 2,000 muscle fibers per every one neuron. Think about your specific muscles within your quads, right? Think vastus medialis, think VMO, think vastus lateralis. I think I said that twice. Rectus femoris, vastus intermedius, right? Since I screwed that up somehow with quads. The 20 to 1 or the smaller motor unit sizes could be things like your hands, right? Where you need more tactile, you need more specific movement, more controlled movement. Or you can have 3 or 4 to 1 in your eyes, trying to control your eye movement specifically. So these finer movements versus gross movements like just like massive flexion extension type movements, right? By increasing the number of motor units that we can recruit and increasing the number of muscle fibers that we recruit, we can increase the amount of force that can be produced, right? So if you think about this, how much force is your eyeball moving going to create compared to a maximal contraction of your quadriceps or maximal hip extension and contraction of the glutes? It's not even close. You can't even compare the two. And that's because larger motor units extend across more muscle fibers contracting, meaning that you get more force. And that's really the hallmark of Hennemans size principle. And this is one of the most important slides that you will see in this entire lecture series. And this is something that you're going to take with you for the rest of your career. And that's as we onset exercise, we're going to start by recruiting the smallest, most minute motor units possible. And then we are going to scale up from there. Our body wants to be efficient. So if it's able to recruit the smallest muscle fibers and complete an action, it's going to. There's no point in recruiting the biggest motor units if the muscle movement requirement is super, super small. Think about, have you ever gone to pick up a box that was loaded down a lot heavier than you thought it was, and you went to pick it up and you couldn't at first? And then as your body and as your brain realizes how heavy it is, you pick it up. That's exactly what's going on with Hennemans size principle. So what these smaller circles are representing is small motor units. And as you increase in size across these motor units, you're actually going to be increasing in contractile potential, or how much force is able to be produced by contraction by going after these bigger and bigger and bigger motor units. I don't know. I love this meme. Sorry. If we're going to talk about motor units and motor control, we got to talk about the brain. I'm not going to lie to you guys. The brain stuff is well outside of my capacity of knowledge. It's super complicated. And realistically, I think that it's incredibly important, but we're going to treat it, we're not going to do it a service. We're going to actually probably do the brain a disservice, unfortunately. So the cerebrum is going to be the largest part of the brain. It's going to be the dome-based structure, essentially, of the brain. It's going to be divided into right and left hemispheres. You've heard of people that are left-brained or right-brained, right? That's going to be talking about the cerebrum. The outermost layer of the cerebrum is going to be the cortex. And for this class specifically, and most of the classes in kinesiology, one of the primary regions of the cerebrum that you're going to be interested in is going to be the motor cortex. So this is going to be the region of the brain that's predominantly in control of purposeful voluntary movement. It would be almost like a misnomer to think that it's where all of the information is coming into. It seems to be that it's the final point at which information is integrated, and then the efferent or the effector signal is sent out from here. The cerebellum is going to be located right above the brain stem. It's going to be used for coordinating and monitoring complex movement. It's the connection between the cerebellum, the motor cortex, spinal cord, brainstem, and spinal cord. So it is a connection center largely. Brain stem, just inside of the base of the skull, just above the spinal cord, if you will. It's largely comprised of medulla, pons, and midbrain. This is going to be a lot of kind of motor control-ish stuff, but it's not necessarily voluntary motor control. Think metabolic functions, cardiorespiratory, so things that we don't have to think about, eye movement, rapid eye movements, muscle tone, how tight your muscles are at specific times, and then as well as the ability to support and combat gravity, so keeping us upright in that. Spinal cord, this is where things actually start getting more and more important for exercise physiology and understanding reflex arcs. So it's essentially, as the signal is sent from the motor cortex down the spinal cord, it goes through a process by which we call spinal tuning. And so if you think about a tuning fork, right, it's essentially creating a generated and a purposeful planned response. So it is a very detail-oriented section. One really cool thing about the spinal cord is it's responsible for reflex arcs. So think about touching a hot object and how fast the signal goes from the piece of your body that was touching the hot object to the central nervous system and then back out, right? Like think about how fast you reflex away from a hot object or away from a cold object. And that's going to be largely because there's a nerve pathway that runs from receptors inside of the skin or in the joint or whatever to the spinal cord, and then it immediately kicks it back to the muscle to say, oh shit, get me away from this, right? It's a rapid response back that's largely predominated by reciprocal inhibition. So that means as the bicep is contracting, the tricep is not being activated, because if you had an activation of both bicep and tricep, you would have co-contraction and your arm would essentially just stick onto the hot thing, which is not what we would want. The autonomic nervous system, we'll talk a little bit about it later on in the semester, but largely for right now, just know that it is the subconscious control. It's the thing that we're not having to consciously think about to control everything. Divided into sympathetic and parasympathetic. Sympathetic is our fight or flight energy system, or our not energy system, but our nervous system. And then parasympathetic is rest and digest, paralyzed. It's a good way to remember it. And they're largely innervating effector organs, so controlling smooth muscle, which would control the rate at which blood flows and the directionality of blood flow. It controls cardiac muscle, the diaphragm contracting down and filling our lungs with air. And most organ systems actually have both innervations. So if you think smooth muscle is actually controlled by sympathetic and parasympathetic, because if you add sympathetic tone to something, you also need to be able to add parasympathetic tone to reverse what it has done at some level. I know I just burned through that. What I really want you guys to take away from this is what are the primary motor systems and nervous systems that are going to be existing? How do reflex arcs work? Really kind of how is a signal being propagated? How's a signal being received with EPSPs and IPSPs? What's depolarization and repolarization? And how's the sodium potassium pump working? Those are kind of the main highlights of this section. If you want later on in the semester, we can dive into brain biology a bit more. We won't really be focusing on it too much throughout the semester, so I kind of just wanted to burn through some of it so that we can keep your capacity a little bit higher for some of the other things. Sound good?